Good morning, everyone. I hope that you're having a wonderful start to your Wednesday. Make sure to get signed into the chat as you come in. Let's get started. Today, we're going to continue talking about genetics, and we're going to focus on gene regulation for the most part. And so just to get started, I'm going to cold call for some definitions. Remember, my rule is that if you do not have an answer that you feel comfortable sharing, you can always pass. That is not a problem. I'm just going to see throughout the class today if I can put a few people on the hot seat just to simulate exam conditions. No stress, a little bit of stress. So let's begin with Kaylee. Do you have a definition for constitutive gene? You're able to unmute Kaylee? Okay, let us go to Alyssa. Do you have a definition for constitutive gene? Got it. Thank you for letting me know, Lisa. Okay. All right, next I'm going to ask Sarah if you have a definition for constitutive gene.
Yeah, so I think like their gene that's clarified all the time, like
That's
they are
right.
open all the time.
Indeed, that's a good definition. It is a gene that is transcribed all of the time. So it's not regulated in the sense of sometimes it's off, sometimes it's on. It's set up so that pretty much it's always being transcribed. And the types of genes that would be set up to be this way would be ones that we would always need no matter what our different circumstances might be just to stay alive. Okay. Repressible gene. What is the definition of this? If you have one, it's fine to pass. Let's ask Andrew first. Andrew, do you have a definition for repressible gene?
A gene that's typically on, but can be turned off when it isn't needed.
That's right. So something that's typically being transcribed, that's its default state, but some kind of change in the environment would signal that it's not needed right then. And then inducible. Bimmy, do you have a definition for inducible?
I think it's when, or this type of gene isn't always needed, but it can be induced based on like environmental conditions.
Yeah, that's a great definition. This is the opposite of repressible. This is a gene that is not normally needed. And so we would have the default state being it's not transcribed, but something in the environment could cue it to be turned on because it's now needed. Last time we talked about how genes in prokaryotes are very commonly organized into operons that are co-regulated by the same promoter. And these typically would be things that are involved in the same metabolic pathway, for example, such as enzymes that are all important to work together to make something, or that are all required to work together to break something. So metabolic and anabolic operons, catabolic and anabolic operons, so making and breaking. So in this little cartoon, we see sort of a typical structure. There's 3 genes here that are all co-regulated by this promoter. And what is the enzyme that binds to promoters in order to begin transcription? Put that into the chat. Yeah. So the enzyme that binds to the promoter is the signal in the DNA sequence that this is the start of a gene, or start of an operon in this case, is RNA polymerase. Now you notice there's this region in between the genes and the promoter for regulated operons and regulated genes. This region is called what? I am going to cold call Aminata. What is this region between the promoter and the genes in a regulated operon?
It is called the repressor.
So you're on the right track. The repressor is the name of the protein that binds this region. So that's absolutely relevant.
The operator.
That's right. Good job. Yes. So you managed to answer both my questions. Good work. So this is the operator sequence bound by the repressor. And the repressor's job is to stop transcription. It's a negative regulation, right? If the repressor is here, even if RNA polymerase is bound to the promoter, it can't go through the repressor. This is a physical barrier, and so we don't get transcription. This repressor protein is encoded by a gene that's usually nearby, usually right next to this operon. And it is a protein that is specific to this operon and is not used for other ones. And you'll notice that this particular gene, sometimes it's called I for inhibitor, but it just depends on the operon and the particular nomenclature used by the people who discovered that particular repressor. It has a promoter that is right next to the gene. There's no operator sequence. There's no way for a repressor to get in between these two areas. And so would you imagine that this repressor is constitutive or repressible or inducible? What do you think? Put that into the chat. Yeah. These types of proteins, these repressors are constitutive. We always need them. Now, this is not to say that they always sit in the operator, because these types of proteins are allosterically regulated. That means that they can change their shape. And when they do so, they can stop binding the operator. So they exist in two forms: the one that's functional or active or bound to the operator, and the one that's not, that has changed its shape and has wandered off. We're going to talk about how a key difference between repressors of different operons is whether or not they're ready when they're first translated to bind the operator or whether they need help. So here we're going to look at a simple example of a repressor that doesn't need any help to bind. So in this example, this particular type of repressor, we transcribe it to mRNA, we translate it into protein, and you see that the shape of this repressor is exactly right to fit into the operator and stop transcription. This is an active repressor, a functional repressor, or what I like to say, this repressor is born ready. It pops out the minute its feet hit the ground. It's like, I know what my job is. I'm going to sit in the operator and stop transcription. Now, does that mean that these genes will never, ever, ever be transcribed? Well, of course not. There must be some way to remove this repressor. These are all repressors are allosterically regulated. That is that their shape can change under the right circumstances. So this type of repressor, there will be some kind of molecule, an inducer, that will change its shape and remove it from the operator, making it possible for transcription to occur. So that's one type of repressor. Now, other repressors are not born ready. They are born lazy. Okay, so let's have a look at that. This type of repressor, once it's transcribed and translated, is not yet in the right shape to bind the operator. And so usually what'll be happening, the default state when this repressor is made, and it's made all the time, and it's just hanging out doing nothing. So there's a whole bunch of repressors just sitting on their mom's couches, eating potato chips and playing video games, not getting a job. Okay, because they're not doing their job, then transcription's happening. That's the default state, transcription's happening. So if your repressor is born lazy, that means that transcription default state is to be transcribed. These guys need a little bit of a kick in the butt. Okay, some kind of environmental cue has to come along, bind them, that causes them to change their shape, or they shape up, and they go and do their job and sit in the operator. And then what happens to transcription? When they're doing their job, when they're active, what happens to transcription? That's right. So what I want you to notice right now that makes understanding this circuit of logic and function tricky is that whatever the repressor is doing, typically the transcription is the opposite. So if the repressor is active and functional and doing its job, transcription is not happening. If the repressor is lazy, non-functional, inactive, not doing its job, then transcription is happening. Okay. So now let's remind ourselves of there's two types of repressors, and that underlies how we can get two different types of operons, repressible and inducible. So let's remind ourselves what the definition of these types of operons is. A repressible one is usually transcribed or not. Transcription is on or off. A repressible operon, the default state is for transcription to be on. That's right. And then something in the environment can turn it off. This is typically how anabolic operons are regulated. For example, the enzymes required to make the amino acid tryptophan. There are 5 genes in this operon, and the enzymes required to make tryptophan take the starting molecule, which is glucose, and change it a little bit. Another enzyme changes that intermediate, a third enzyme changes the next intermediate, until finally we get tryptophan, the final product, which is an amino acid. Now, amino acids are really important. You have to have them all the time. So you might have assumed that this type of thing would be constitutive. But what sort of circumstance do you think would be one where we would want to stop making tryptophan? Is there an environmental cue that could trigger that? Yeah. What if a bacteria ran into a turkey leg or some source that was tryptophan rich? Okay, so suddenly in the environment, there's a whole lot of tryptophan. Does it make sense to be diverting a significant amount of energy, because this type of synthetic process, this building or anabolic process, requires energy? Does it make sense to continue pouring energy into production of something that you already have lots of? Or does it make sense to kind of put the brakes on that so that you can use up the environmental source and then go back to making it later when you've run out? So it makes sense then that the environmental cue would be high tryptophan. And then when the tryptophan levels drop, we go back to our default state of transcription. Let me just back up for a second and ask you this question then. If we want our default state to be transcription is happening, what kind of repressor do we need? Do we want one that's born lazy and won't get in the way? Or do we want one that's born ready and is oh so willing to do its job and is in the way? What do you think makes more sense? If we want to engineer something so that the default state with no input is transcription is happening, and only when tryptophan levels rise do we turn it off? Do we want our repressor to be born lazy and just kind of laissez-faire, and only do its job if the environment says so? Or do we want it to be born ready and not need any help? Yeah, you guys are right on the money. We want our repressor to be born lazy, okay? So as it turns out, in the greater scheme of things, in the greater good, in this analogy, laying on the couch eating potato chips and playing video games might be useful to society, okay? Let's talk about inducible operons. Now, an inducible operon is one where the transcription default status is what? That's right. In an inducible operon, transcription is off just as a default with no further input. That's like the default state. Something in the environment can turn transcription on. This is typically how we regulate things like unusual carbon sources. Maybe unusual is the wrong word. maybe non-optimal is a better word. Carbon sources that are not as good as the best one. Let's just say, let's get the elephant out of the room. What's the best carbon source in most cases in cellular biology? That's right. The best carbon source is glucose. And the reason is that cells are set up typically to use glucose. And so if you want to use lactose or sucrose or galactose or fructose or xylitol or any of the other possible sugars, you have got to put a little money in up front to convert those sugars into something that actually can fit into the pathway that's optimized for glucose. So you're just never going to get as good of a yield. And so for the metabolizing these types of things, we usually don't make them unless what? Thinking specifically about the example of the lactose operon, the enzymes required to metabolize lactose, what should be the environmental cue to cause those enzymes to be made? Yeah. So an environmental cue that would be very useful is lactose is actually present. We don't want to be wasting time and energy and ATP making enzymes for something that is not around. So the presence of lactose is really key. Some of you have additionally alluded to the fact that in these types of operons, there is a second level of regulation, that there's not just the substrate that's important, but also the presence or absence of glucose. So to be very complete, we could say that for these types of inducible operons, what we need is for the substrate, lactose, for example, to be present and glucose to be absent. So let's see if we understood. Oh, wait, let me just go back and ask this question, which I forgot to do, I'm sorry. If what we want is an inducible situation where usually transcription is not happening, what kind of repressor do we want? Do we want one that's born ready and is sitting in the operator in the way? Or do we want one that's born lazy and is kind of hands off? That's right. If what we want to accomplish is a default situation where transcription is not happening, then the best way to do that is to have a repressor that's born ready. Okay, so let's see if we understand that based on these pictures. Let's have a look. This is an example of a gene. Could be an operon with multiple genes, but let's keep it simple, one gene. There's the promoter to which RNA polymerase is bound. Here is the operator. And you can see that this constitutively made repressor is born ready, bound to the operator right out of the gate with no further input. If we want to remove this repression so that we can allow transcription, we have to have some kind of environmental cue that binds the repressor, changes its shape, and takes it off the operator. Does this describe an inducible or repressible type of regulation? Yeah. Inducible, right? Because what's the default state? The default state is there's no transcription. And that's what inducible means. Is there's the default state, without any further information, what is the cell going to do? It's going to say, nope, we're not making you today because the repressor that's born ready is in the way. If we want to make this transcription thing happen, we've got to take the repressor off, and that's going to require some kind of environmental input. Okay, let's look at this situation. Now we have a gene. Here's our promoter. Here's our operator. Here's our constitutively made repressor that's always around, but you'll notice that it is not in the right shape to bind the operator. It is born lazy. It's hanging out on the couch, all relaxed, drinking soda, talking on the phone. If we want it to do its job, we've got some kind of environmental signal has to come along and get it into shape and tell it to sit down and do what it's supposed to do. So the default state is transcription is happening. Is this repressible or inducible? Yeah, exactly. This is known as repressible. All right, let's talk about the tryptophan operon. There's one of two operons that you're responsible for that are sort of case studies, but I will also expect you to understand the principle, like how to identify if an operon is repressible or inducible based on the information. So let's do that now with the tryptophan operon. Even if you've done your prep, you already know which one this is, but that can be hard to remember too. So let's talk about it. This is an anabolic operon, meaning the genes are codes for enzymes that make stuff. In this case, in the tryptophan operon, the genes make the enzymes that are required to synthesize the amino acid tryptophan from the starting material, glucose. Remember that this process costs ATP. So tryptophan is something that we definitely need lots of supplies of all the time, because we never know when we're going to have to make a protein with tryptophan in it. So we can't be in a position where we run out. Therefore, the default state for this operon is that it's transcribed. And an environmental condition can turn it off. So if suddenly E. coli runs into a whole stash of tryptophan, what should it do? Yeah, turn off transcription. Pump the brakes on making more tryptophan. So if that's what we want to accomplish, then are we trying to set up an operon that is repressible or inducible? Where the default state is we're making the enzymes to make tryptophan, and then the environmental cue, tryptophan itself, turns transcription off. Correct. This is repressible. And in order to accomplish this, when the repressor is made, should it be born ready or lazy? Should it be born active or inactive? Should it be born functional or non-functional? Do we want it on the couch or do we want it at the Safeway? Great. Yeah, you want a nice sedentary, laissez-faire, devil-may-care repressor that's got its feet up on the couch. So here is a diagram of that. Here are the 5 genes in the tryptophan operon. Here's the operator where the repressor will bind, the promoter where the RNA polymerase will bind. This is the sequence that encodes the repressor itself. It's constitutively made from a different promoter. And you can see that it's always made in this initially inactive state. And so consequently, transcription is always happening. Enzymes to make tryptophan are always being made. And so tryptophan pools are rising. What is the environmental cue that is going to turn the transcription event off? Yeah, high levels of tryptophan. It could be environmental, or it could be that the cell's been making a whole bunch of tryptophan, but it hasn't been needing any tryptophan in the proteins it's made lately, and so there's extra tryptophan laying around. Either of those would signal high levels of tryptophan, and that would cause transcription to halt. And how does it actually work? What does tryptophan actually do to accomplish that? Yes, Nicole. Tryptophan acts as the co-repressor. It is the allosteric regulator of the repressor. It binds the repressor and tells it, go get a job, you lazy slob, do something useful with your life. And then the repressor suddenly realizes, that life has meaning and goes and does its job of sitting in the operator. And consequently, transcription stops. Okay, so when tryptophan is not around, what is the status of the repressor? Is it active or inactive? That's right. When tryptophan is not around, the repressor is not active. And as a consequence, what's happening with transcription? Correct. Transcription is happening. Add some tryptophan to the scenario. Okay. Now what is the repressor doing? Yeah, the repressor's been activated. It goes and sits in the operator. And what happens to transcription? It turns off. That's right. So you can see that happening here. Here's the default state, lazy repressor, lots of transcription. And then environmental tryptophan or high levels of tryptophan cause the repressor to bind the operator. Changes its shape, it binds the operator, transcription stops. And just to add this level of nuance, this is not a permanent situation. This binding between tryptophan and the repressor is not like glue. It's like a hug between people that really like each other. And occasionally one of them takes a bathroom break or goes for a coffee or whatever you want to think about it as. And just for a second, it goes away. Now, if there's a lot of tryptophan still in the cell, then while this one's on a break, another one's just going to pop right in there and this guy won't even have a chance to leave. But if this guy pops off for a second, and meanwhile we've had a drop in tryptophan and there's a ribosome there going, hey, yo, I need a tryptophan right here, and that tryptophan gets co-opted into protein synthesis, then this repressor is left with no one to touch, right? And so it reverts to its lazy conformation. And then we get transcription again. So that's the first operon we have to know about, and also just how to identify a repressible operon. How does it generally work? Our next case study is the lactose operon. So the lactose operon has 3 genes shown here, permease and an enzyme to break lactose, the disaccharide, into its component glucoses and galactose. And it's got the operator, where the repressor will bind, the promoter, where RNA polymerase will bind, and this gene that encodes the repressor for lactose only that binds to the lactose operator. These enzymes encoded by this operon are what are required for lactose metabolism. And so again, we only want them to be expressed under specific environmental conditions. And we don't actually want them expressed most of the time. So the default state for this operon is that it is not transcribed. And some kind of environmental change, the presence of lactose being part of that, causes this transcription. So are we talking about something that we want to set up to be inducible or that we want to set up to be repressible? Right. This is inducible. If what we want to do is set it up so that it's inducible, so that the default state is there's no transcription and then some kind of environmental change can relieve that repression, then do we want our constitutively made repressor to be born ready or born lazy, born functional or non-functional, born active or inactive? What do we want? Yeah, we want that type A firstborn child, eager to please, putting their boots on the ground, barely out of diapers, off to get a job at the local bank or whatever. We want a repressor that doesn't need any help to do its job and sit in the operator. And when that happens, that means the transcription cannot proceed. So here is the lac operon. In its default state with no environmental input, because the repressor is made in the active conformation and sits immediately into the operator. and then we can't get transcription, which is good news, right? What should cause this repressor to go away? The inducer, the presence of lactose. Okay, and this is shows you how that works. So lactose is now an inducer. It binds the active repressor, inactivates it, so it can't bind to the operator anymore. And that allows the possibility for transcription to occur and for these genes required for lactose metabolism to be expressed. So we'll just review that first. When lactose is not around, no lactose in the environment, the repressor is active or inactive. That's right. When lactose is not around, the repressor is active, and consequently transcription indeed, transcription is off. Good. So we have our default state that we wanted to achieve. Now let's add some lactose. What happens to our repressor? It's sensitive to lactose. Yep, it becomes inactive. Exactly. However, that does not automatically mean that transcription turns on. This is more complicated than the tryptophan operon. And that is because lactose isn't the only stakeholder in this scenario. We also have to take into account whether or not what else is true in the environment. Besides lactose, what else is going to matter? We also need for glucose to be absent. Now, I will give you an opportunity to raise your hand and explain why that is important before I do. So if you've done your prep, maybe if you've already been to lab, maybe you'll know kind of how you think about this. So give me your best shot at it, Erica.
I think I may be getting ahead of where we are right now, but does it have to do with like the levels of glucose and ATP?
It does, yeah.
Okay,
Exactly.
so if glucose is low, ATP is low, which means it'll bind the cap, and then yeah, that's pretty much where I'm going with that.
Yeah, and CAP is this other thing that we haven't talked about yet, but is really key to the lactose operon and to many operons like it. Anyone else want to comment on this before I go through my own paces? Something you'd like to add? Nicole, go ahead. What would you like to add?
So Erica said pretty much most of it. But so for inducible operons, you need to have this cap binding site area, which is kind of like you described as a light show that says, hey, to the RNA polymerase to come here, because otherwise it wouldn't know where to go, because these operons aren't usually on. So that needs to happen, but in order for that to happen, glucose needs to be absent, because if there's not a lot of glucose, that means there's a lower ATP yield, which means that, and then for some reason it's what's the word, inversely related to this thing called CAMP. And so if there's low ATP, then there's high CAMP. And what the CAMP does is it activates the CAP molecule. And so what the CAP molecule will do is bind to that light show area, turning it on so that RNA polymerase actually knows where to go, so that, because if it doesn't know where to go, then transcription doesn't happen.
Yes.
So yeah.
Yeah, that's a great description of how CAP works and how glucose is involved in determining whether CAP is there or not. And so if you're feeling a little confused, don't worry, because we will go through that again. But that's a great synopsis from the prep that that I asked you guys to do. So before I go into reviewing those details, I do want to convince you that this is actually important. And I've talked about this a little bit before, but and if you've been to lab, you've heard this before, but I just want to reiterate it. So I want you to imagine that there are, well, first of all, we have to accept this. I said this earlier, but I need to say it again because you might not remember it or believe me yet. But of all of the carbon sources, what's the best one? Yeah, by now I've indoctrinated you into thinking that glucose is the best carbon source, and that's excellent. The reason for that, again, is that the cell is set up to get energy out of glucose. That's what the machinery is all for. So if you want to use something else, you have to do something to convert that molecule in a way that's going to lose or cost you energy. So glucose is the best carbon source for that reason. Okay, so if we accept that, then exactly, Mariceli, CAP is what allows cells to be picky. Yes, Nicole said it very well. Yep, good job. Okay, so imagine two cells, okay, that are living in an environment where there's glucose and other carbon sources. We'll call one of them Bob, and we'll call one of them Stanley. And Bob is not a picky eater, and Stanley is, by which I mean selective, perhaps would be a better word. So both of them are capable of using glucose and the other carbon sources, but Bob doesn't really care which one he uses first. If he's living in a soup of glucose plus lactose, he uses a molecule of glucose, a molecule of lactose, a molecule of glucose. You know, it's just whatever happens to be the closest to hand at that particular moment. And so he gets, you know, a burst of energy with glucose, a little bit less with lactose, a burst of energy with glucose, a little bit less with lactose. That ATP is driving his cell division. So he's got sort of a moderate amount of cell division going on. He has a decent number of kids. He's pretty content. Over here, we've got Stanley, who's like, I only take the best when it's available. And so he's living in the same world, glucose plus lactose, but he is selectively picking only the glucose. So for each unit of time, he's getting a burst of energy, a burst of energy, a burst of energy, a burst of energy, and he's using that energy to make more kids and more kids and more kids and more kids. Who is winning the evolutionary race if winning means you have more kids? Yeah, Stanley, the guy who's selective. That's right. So there's a strong evolutionary pressure to be selective. When both sugars are present, actually, if you're able to ignore things that aren't glucose, if glucose is around, then you're going to be more successful. And so we need to talk about how that works, because repressors alone are not going to accomplish that. Okay, let's have a look at our time. We're not taking our break yet. It's coming up. So again, we have our situation here where these genes are required, or the product of these lactose operon genes are required to metabolize lactose. One's a permease, bringing lactose in, one's an enzyme, chopping it in half. And we only want these to be expressed under what environmental conditions? Lactose present, glucose absent. That's right, because that's going to give us the most efficient use of resources, and it's going to allow us to win the evolutionary race. So how do we accomplish that? This picture that I have painted, I have to add a detail to it that involves CAP. Some of you already know where I'm going with this, but let me just sort of revisit this idea, but add in the extra information. When lactose is not around, the repressor is active, that's its default state, and transcription is off. That's still true. When you add in lactose, it still binds the repressor and inactivates it. That's still true. Okay, so the repressor is sensitive to lactose. That's still true. If there's no lactose, the repressor is bound to the operon at the operator location. And if lactose comes along, the repressor does go away. Okay, so that doesn't explain how glucose gets to have a say in all of this. What Nicole was getting at and what Erica was getting at is that the reason we can have glucose have a say in this is that the promoter of the lactose operon, and this is true of inducible operons in general that have a additional regulation, the promoter is not very good. Okay, RNA polymerase doesn't normally bind to it. There has to be some kind of additional activator. The activator is what is going to be sensitive to glucose levels in order to get RNA polymerase there. So even if you take the cow off the tracks or the boulder off the tracks, even if you remove the impediment to transcription with lactose, it's not enough because the promoter is not very good. So we are not going to get RNA polymerase there unless we have this activator. So activators are the opposite of repressors. Instead of exerting negative control, they exert positive control. Just like the repressor, the activators bind to the innate control regions, and they basically, make that promoter visible. They turn the lights on at the airport so that the planes can land, is my favorite analogy, but they make it possible for RNA polymerase to bind to the promoter. And the one we're going to talk about is CAP. CAP is one of these transcriptional activators, and it is used for many catabolic operons that are regulated in an inducible manner, ones where the genes make enzymes to metabolize non-optimal carbon sources, ones where those carbon sources are ones we would like to ignore if glucose is around. Okay, this is what enables E. coli and other microbes to preferentially use glucose. Even if other carbon sources are available, use glucose first and switch to those only when glucose is gone. So here's the true picture of the lactose operon. And I'm going to describe it for you right before the break. We're going to go through this in pictures in a number of ways. So what we have here is our genes for, these are not actually called A, B, and C. They're actually ZYA, but there's 3 genes in the lactose operon. And we've got our promoter, our operator, and now we've got our repressor gene, and then we have this additional site, this CAP binding site. So just so we can refresh our memories about everything, again, what binds to the promoter region? Put that into the chat. What binds to the promoter region? Yes. The promoter is where RNA polymerase binds, and it's going to try, when it binds, it's going to try to transcribe these genes. But why is this CAP binding site necessary? Is this promoter an awesome promoter like the constitutive promoters are, or like the tryptophan operon promoter is, where RNA polymerase just binds as soon as it sees it? No, this is a crappy promoter. It's not very good. Nicole?
So I just have a question, and I want to see, because this is how I understand how RNA finds it. So for repressible operons, those are the ones that are going on like all the time. So RNA polymerase is like used to those. So it's like he knows that those ones need him all the time, but these ones are only turned on once in a while. And so he doesn't really care about them or know that they're like there. So he needs the CAP.
Yeah.
Is that right?
Yeah, that's a good way to think about it. I will give you a more mechanistic, accurate way, like in terms of actually like molecularly, what's the difference?
Mm-hmm.
Remember what the promoter actually is it's just a series of letters in the DNA. And actually, and this is beyond the scope of this class, but I'll just mention it, like the series of letters in the DNA that make a promoter sequence, there's not just one way to spell promoter, basically. It's a variety of things that have a certain kind of pattern. They have a certain sort of ratio of GCs that RNA polymerase recognizes. And so some of those promoters are just extremely easy, or places where RNA polymerase will bind very strongly because of literally because of the series of letters. The spelling of promoter is very obvious.
Okay.
And other promoters, like the ones in the inducible operons, they're kind of misspelled. Like the letters are kind of there, but some of them are a little bit obscured. Like, you don't have quite the right ratios of GCs, Ts, and As to make it a really appealing place to land or a really obvious place to land. And so RNA polymerase is actually not able to bind it very well, is what it comes down to. It's like a physical interaction. And so
Okay.
just the same way like an enzyme binds its substrate, like a antibody binds its antigen, that fit is not very good. And so the analogy I always use is that it's kind of like, it's like a dark airfield and this is the lights, and that's a good analogy. But what is really happening is that CAP is also kind of tugging on RNA. It kind of takes it by the arm and says, this is where I'm sitting down, you're coming with me.
Okay, that helps a lot. Thank you.
Okay, so RNA polymerase binds the promoter, but in inducible operons, this promoter is kind of crappy, so it's not going to bind by itself. What it needs to bind here, if it's going to, is that CAP also needs to be in the CAP binding site. Meanwhile, we've got this gene, which is the repressor, and where does this, and this is constitutively made, as we can see, there's no operator in between the gene and the promoter for that gene. And when it's made, it goes and sits in the operator. The negative regulator is the repressor, and this is its default state, okay? And the positive regulator, okay, is because this promoter is not very good, RNA polymerase is not here by default, actually. It only shows up if also what's true. For RNA polymerase to be here, what else has to be true at this operon? Yeah. We also need CAP, the activator, to be here to bring RNA polymerase here. And what's important to recognize when you look at this picture is that these two proteins, the negative regulator repressor and the positive regulator CAP, are similar in that they're DNA binding proteins that have a very specific place to bind, and they are allosterically regulated, which means they have another shape. And if they change into this other shape, they'll go away. And that, whether they're bound or not bound, is dependent on a specific environmental cue, but it's a different cue. So for the repressor of the lactose operon, the repressor is sensitive to what? What could bind the repressor and make it go away? For the lactose operon, what is this repressor sensitive to? If it bound to that thing, it would go away. Yeah, the inducer. And what is the inducer? The inducer is lactose. That's right. Now, if this was a different inducible operon, let's say arabinose, then that would be a different repressor with the same function of binding here and with the same property of being active right out of the gate. But the arabinose repressor would be sensitive to arabinose instead of lactose. CAP, on the other hand, is universal, and it's sensitive to glucose levels, but it's not direct, because as both Nicole and Erica kind of alluded to, there is a sort of series of steps that gets to this, and we'll talk about that shortly. Okay, let me just check my time. Okay, I'm going to talk about what controls CAP, and then we'll take our break. So what controls CAP? Some of you already know this. CAP is sensitive to what? It's not sensitive to lactose or arabinose or anything like that. And it's not directly sensitive to glucose, although it is affected by glucose. Glucose levels control CAP, but not physically. What directly physically binds to CAP and determines whether or not it's going to be active? Right. That's right. Cyclic AMP. Cyclic AMP is a small molecule that has that does a lot of things in the cell. You may have heard of it before in physiology, but in this particular case, this protein is controlled by cyclic AMP. And what that means is, in order for CAP to bind to the CAP binding site, cyclic AMP has to bind to it first. So cyclic AMP has to convince CAP to be active. And so how does that help us to sort of involve glucose? Well, cyclic AMP is affected by glucose levels. And the reason for this is that cyclic AMP is metabolically related to ATP in an inverse relationship. ATP and cyclic AMP both are made from the same starting molecule. So when ATP levels are high, cyclic AMP levels are low, because basically ATP is hugging the starting molecule. And when ATP levels drop, there's more stuff to make into cyclic AMP, right? So they're inversely related. Now, when there's lots of glucose, remember, it's a really good carbon source. So if glucose is high, what do you think ATP levels are doing? When glucose is high, ATP is going to be high, right? Glucose high means lots of glucose in the environment means we're making tons of ATP because this is a really good carbon source. Since ATP levels are high, cyclic AMP levels, on the other hand, are going to be low. Okay, that is how glucose is related to cyclic AMP. And if cyclic AMP is low and it's not binding to CAP, is CAP active or not? Not active. Okay, so high glucose means CAP is resting. I think a useful way to think about CAP is that CAP is kind of like an emergency fire marshal, right? Or a first responder, like, you know, maybe a fireman. So if there's no emergency and having lots of glucose means life is cushy, then it's just resting, like it's, you know, cleaning the wheels of the bus or whatever. But if we run out of glucose, that's an emergency. We've run out of our best carbon source. We basically could die. So we better do something about that. And that's when CAP gets activated and starts running all over the place, trying to motivate the genome to make something that would be useful. Glucose high, CAP's not active, not bound. So that means that RNA polymerase is not at the crappy promoter, and then we are not getting transcription. Now let's glucose run out. ATP levels go. Glucose drops, ATP levels drop. Cyclic AMP, on the other hand, shoots through the roof. What happens to CAP? Yeah, the high cyclic AMP is like the alarm, and CAP is like, holy shit, what's going down? We've run out of glucose. We might die. Let's do something about that. And it runs all over the genome and sits down in all these CAP binding sites for all these non-optimal carbon sources, bringing RNA polymerase there, just hoping that at least one of those carbon sources is actually present in the environment and able to relieve the repression so we can get transcription of those genes so we don't die. Okay, so when glucose is low, CAP gets activated, it gets alarmed, it runs and binds to the CAP binding site. It brings RNA polymerase with it and says, hey, buddy, you know, like, I don't care that you don't normally see this promoter, but this is an emergency, like get down here. Let's see if we can get some work done. And then whether or not that transcription is successful depends on the repressor, which is sensitive to that particular carbon source. So with that, we'll take our break. I'll see you at 11:05. All right. So let's walk through these different scenarios so that we can better understand how these environmental sugars affect transcription of an inducible operon, such as the lactose operon. So if there is neither glucose nor lactose in the environment, the fact that there's no lactose means that the repressor, which is sensitive to lactose, and if lactose were present, it would be a different shape and unable to find the operator. If there's no lactose, is the repressor bound to the operator or not? Right. Because there's no lactose, we have just our default state. The repressor is bound. And also, if there's no glucose, then ATP levels are low. Consequently, cyclic AMP levels are high. The alarm is sounded. CAP gets all upset. It grabs RNA polymerase, says, hey, dude, come look at this promoter you've been ignoring. But even though it's present, and RNA polymerase is present and it's ready, the repressor is in the way. So do we get transcription? We don't. Now, is this what we want? Like, does this make sense? Does it make sense if there's no lactose and no glucose for CAP to have brought RNA polymerase just in case, because we're in an emergency situation, but since there's no lactose, the repressor is still there and we're not wasting our precious resources making something that's not going to help us. Does that make sense? Great. So we've achieved our goal. Okay, so what if we still have no lactose, but we add glucose? Okay, so no lactose means the repressor is bound or not bound. Yeah, no lactose, repressor still bound. We've added glucose. So ATP levels went up, right? What happened to cyclic AMP levels? They went down. The alarm turned off. CAP said, oh, panic over, glucose is here, thank God. And it went and took a break. And RNA polymerase, therefore, is here or not here at the promoter. That's right, RNA polymerase is not here. Okay, so the repressor is still in place, but since there's not even an emergency, CAP isn't even there with RNA polymerase. Like there's no possibility of transcription in this scenario. Transcription's still off. Again, is this what we want? Sure, because first of all, there's no lactose. We definitely don't want to make these genes, but furthermore, glucose is present. So we don't even we're not even in a situation where even if lactose was around, we'd want to make it. Okay. Now we keep the glucose, right? Glucose is still present, lots of glucose. Glucose is everywhere. Let's add in some lactose. Because glucose is present, ATP levels remain high. Cyclic AMP levels remain low. The alarm does not sound. CAP is still relaxing. RNA polymerase here or not here? Not here. Okay. So there's no RNA polymerase. Lactose is present, however. So what's happened with the repressor? Yeah, lactose binds the repressor and sends it away. Now, we still don't get transcription, right? Because there's no RNA polymerase because glucose is around. But if glucose were to run out, we'd be kind of positioned for transcription to happen, wouldn't we? And so let's talk about that circumstance where we actually do get transcription. What is that environmental condition again that we want to be true before we bother to make these genes? That's right. We want lactose to be present, but glucose to be gone. So let's suppose we got both sugars and we eat up all the glucose and only lactose is left. Okay, so because lactose is left, it has already disabled the repressor. And because glucose was used up, ATP levels went down. That caused cyclic AMP levels to go up. And remember, cyclic AMP is like an alarm. It's like a sound. It goes up, whoop, 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 and cap is like, oh no. And so it comes and binds the cap binding site and brings RNA polymerase with it. And so now can we get transcription? Finally, we get transcription. Amazing. So does that make sense? Yes, no, kinda. Great. So these slides are posted in the test 2 prep page in week 6. No, week 7. One of the weeks, the week right before the test. And this recording is going to post, of course. So I encourage you to review it. But here's the most important thing you can do for yourself. Draw this picture, okay? And diagram what happens in the scenarios. But remember that the repressor for this operon is sensitive to this sugar. And CAP cares about glucose. And you should consider them separately. It is unhelpful for most people who have just regular intelligence and are not like super geniuses to try to solve a logic problem all at once in their head. A picture is very important. And taking each piece of the logic puzzle separately and saying, well, what is the repressor sensitive to? It's sensitive to lactose. Is lactose present? Yes or no? What's happening then? And what's CAP sensitive to? Is glucose present? Yes or no? What's happening then? Instead of trying to just solve it all at once with no picture in your head, it's going to be difficult. Okay, so next we have to talk about something called catabolite repression. And this is just a fancy way of saying glucose is used before other stuff. But I want to tell you how we found out about this, because it was not actually intuitive. People didn't realize this was going on. They saw some data that was very puzzling and had to figure it out. So I want you to just take a second to return to that state of innocence before I told you that glucose was the better carbon source, and to just make the null hypothesis that all carbon sources are sort of equally valid. Let's take a species and grow it into 2 different broths. We're going to grow that species first in a glucose broth, and we're going to see this growth curve. This is the log phase of the growth curve. We're going to take that same species and grow it in lactose only, and we can see that this is the growth curve. In which of these conditions is the species growing the fastest? That's right, in glucose. And in which of these conditions is the generation time the shortest? I ask it that way because it is not always immediately obvious that that's the same question. So if you're growing fastest, meaning your cell divisions are happening more rapidly, your generation time is going to be shorter. Okay, so glucose is the answer to both those questions. If we were to assume that cells are not selective, right? That if we put them into a medium that has both sugars, they're going to eat a little bit from column A, a little bit from column B. They're going to kind of mix and match like they're at a wine and cheese, like a little bit of brie, a little bit of cheddar makes everything better. Okay. If we were to assume that that was the case, which was the initial assumption, and we put this species that does really well in glucose and not so well in lactose into a broth with both sugars, if they're eating them simultaneously, then you might expect that the generation time would sort of be intermediate, right? And we would see a growth curve that kind of runs up the middle, kind of like this. But that is not what happened. When you take something like E. coli and you stick it into glucose plus lactose together, you instead see something that looks more like a ladder. We see first a growth curve that looks like the glucose growth curve, then we see like a flat line, and then we see a growth curve that looks like the lactose growth curve. First of all, what happened at this inflection point, do you think? Yeah, lots of great answers in the chat. Not everyone answered this the same way, but you all answered it correctly. So basically here is where we run out of, we ate up all the glucose. That sent us, means we're now in a new environment. We have to adapt to that new environment. We have not run out of food, right? Because what's left? Lactose. So we're not going to enter a stationary phase. What we're going to do is enter a lag phase where we have to change what enzymes we have. We've been, using glucose, which the cell is already poised to do. And then boom, we run out of glucose, but lactose is still present. Now we got to switch to making the lactose metabolism enzymes. That's going to take a minute. Then we take off again. By the way, if there was then another flat line over here on the right-hand side, what part of the growth curve would that be? Yeah. If the only two carbon sources are glucose and lactose, and we first run out of glucose, then we switch and we use lactose, at some point we're going to run out of lactose and then we'll hit stationary phase for real. Okay. So let's ask some questions. So here's a growth curve with a little bit more detail in it of E. coli that's plunked into a glucose plus lactose broth. And we initially see that there's no change in cell number. Then we see a very sharp rise in cell number. Then we see a flat line again, and then we see a shallow curve. If we were to name the parts of the growth curve here, which of these would be the best, most accurate nomenclature to use? This phase is what, this phase is what, and so on. Yep. So first we add these cells to a new environment. They have to adapt. That takes a minute. Then they take off running and they eat up all the glucose. Then they're in the new environment with just lactose. They have to adapt. They eat up all the lactose. Boom. If we had another flat line, we'd be in stationary phase, but we are lag log. Okay, now let me ask you this question. And before I do, I have to just make sure that we are on the same page about one of the terms in these answers. So beta-galactosidase is an enzyme, and it's one of the enzymes that is made when transcription of the lactose operon is happening. So it's one of those three, it's LAC-Z, that gene LAC-Z codes for beta-galactosidase. So this is going to be made when transcription of the lactose operon is happening, and it's going to not be made when transcription is not happening. Okay, so here we are. In this graph, A is the label for the steep curve and B is the label for the shallow curve. And if we look at these options, which one is true when we are in A? So read your options, see if you can figure it out, put your answer into the chat. Great. Okay, let's talk about it. So great job, you guys. None of you got the right answer, but that's really common the first time that I ask this question. So what I want to do now is just talk about it a little bit and see if we can arrive at what that correct answer is. All right, so let's first think about what is going on here. The cells are in an environment that contains both glucose and lactose at the same time. Let's just think about the lactose part of it first. Lactose is present. So what's happening? Is the repressor doing its job and bound to the operator, or is it not? what's the repressor doing? Remember, the repressor is sensitive to lactose. And lactose is around. So lactose has bound the repressor, but that means the repressor is now relaxing, taking a coffee break, pouring some cream in its coffee. And so it's not bound. So that kind of eliminates a couple of these options, doesn't it? Kind of eliminates C and D right out of the gate. And that leaves us with 2, or maybe E as well. Okay. Now let's think about glucose. Glucose determines whether or not CAP is bound to the operon. Since glucose is present, ATP levels are high, right? Cyclic AMP levels are therefore low. There's no alarm. CAP is relaxing, right? It's like coiling the hose on the fire truck. It's a nice downtime. No one's had any sort of emergency, maybe playing cards. So CAP is not bound to the operon. Because CAP's not there, is RNA polymerase there? No. So is transcription happening? No. So is beta gal being made? No. So what's the answer, guys? Right. Now, I want you to take a second right now to reflect on how you originally got to the answer you got to, which was incorrect. Think about what the reasoning was that you used. Were you just kind of like reading the question and kind of skimming through it? Were you trying to solve it all in your head? What's the missing piece so that you can get this right? I want you to think about that. Did you need to slow down? Did you need to go, lactose is doing what to the repressor? Glucose is doing what to the CAP? Like whatever that process is, okay? Think about how you can get there so that you can be successful on the application questions on my tests. Okay, great. Good work. Let's go to the next one. Now we're in B. So what's true? Think about that, put your answers in. Don't be afraid to guess. Don't be afraid to not know. This is the time for learning, but do your best. Great. Okay, this time most of you got the correct answer, but let's just talk about it. So in B, what is present in the environment? That's correct, lactose only. So lactose is present, meaning the repressor is bound or not bound to the operator. Not bound, okay. Glucose is absent. ATP levels went down, cyclic AMP levels went up. CAP is therefore CAP is active, right? And it's bound to the operon. And so what's happening with transcription? It's on. Right. Good job. And this is exactly what we would want or predict, right? Because if we are going to use lactose, we better have a way to use it. We need the enzyme beta-gal to be made. Okay. Now, remember I asked you, what if there was another flat line at the end here? What if we went into stationary phase at the end? Which of these would then be true? Think about it, put your answer into the chat. All right, let's talk about it. So now what's true in the environment in stationary phase? Do we have glucose or lactose? We have neither. Okay. So let's start with lactose. Lactose is gone. Is the repressor bound or not bound? Yeah. Lactose is gone, repressor is bound. Glucose is gone. That means ATP levels went down, cyclic AMP levels went up, the alarm went off, CAP is activated and it goes and binds to the operon. So far, that means the best answer is D, but wait. But wait. If CAP is there, but the repressor is also there, are we getting transcription? Yeah, even though CAP's hair is on fire and it's like emergency, emergency, ran out of glucose, and it's bringing, remember, it's binding all over the place because there's more than one place in the genome to bind. All the inducible operons have a CAP binding site, and it's bringing RNA polymerase there and it's sitting down. But in this particular case, there's no lactose. So the repressor is in the way and it's therefore not able to transcribe. So beta-gal is not made, which is exactly what we don't want to spend our last energy making something we're not going to use. So what's the right answer? That's right. It's E. And that is a reminder to A, read the question carefully, right? And B, that I don't have trivial answers to things, right? If there's a none of the above, you should take it seriously as a possibility. Okay. Let's talk about this scenario. Let's suppose that E. coli developed a mutation in the lactose repressor. It changed it in a way that it couldn't bind lactose. So just remember this. Remember that the repressor is an allosterically regulated protein. That means that it's got a place to bind DNA, but it has this other little spot that lactose fits into, right? And that's required for it to change its shape and then leave the operator. So if we were to mutate the amino acids in that little spot where lactose binds, and now lactose can't bind, which of these answers do you think would be the most likely outcome? Yes, John, that's funny. Y'all are correct. D is the correct answer. Raise your hand if you can explain why. All right, I'm just going to cold call a couple people from the chat. If you, the reason you chose D was it was a total guess, then that's fine to say. Or if you have a reason you don't feel very confident about, but you're willing to share, then that's fine. And if you're not willing to share, you can always pass. But let's just interview some people. Let's start with you, Stephanie. Why did you say D?
Because it would never be taken off the tracks because lactose wasn't there.
Yeah, that's a good
Take it
answer.
off.
Yeah, this lactose repressor is born ready. The only way it'll come off is if lactose binds to it. If it's not able to bind lactose, then it just stays ready forever. It never gets to take a coffee break. No creamer in the coffee, right? It's just standing there for its whole life until it's dead. Okay. Let's go through all these fill in the blanks real quick. And I think I'm just going to cold call people for this. Lactose operon, is it anabolic or catabolic? Braden.
I think I'm gonna pass.
That is not a problem. Let's go to Courtney. What do you think? Lactose operon, anabolic or catabolic?
I'm gonna say catabolic.
Yes. Okay. And then lactose operon repressible or inducible, Emily.
I'm gonna guess and say reducible.
Inducible is correct. All right, Erica Lowry, the environmental signal, lactose, acts as a what? Inducer or co-repressor? It looks like Erica is not able to unmute. Alina Park, are you able to tell us what you think is the environmental signal lactose, an inducer or a co-repressor?
I would pass.
That's no problem. And Erica just said she passed too. Thank you for letting me know, Erica. That's excellent. Jordan, what would you say?
I'm gonna pass as well.
Not a problem. Let's go to Mai. What do you think?
Is it inducer?
It is an inducer, right? That's good. All right, now by default, what state is the repressor in, active or inactive? Let's go to Mabintu.
Is it active?
By default, it's active. That's correct. And then the default state of transcription, Mariceli, is on or off?
I would guess it's on.
It is off. It is off. But I think what you're thinking about is that the repressor is bound. Is that what you're thinking about?
Yeah.
Yeah, that's what I thought. I could hear it. All right, good job, everyone. Let's go to the tryptophan operon. Tryptophan operon. Is it catabolic or anabolic? Melissa.
I'm gonna pass.
Not a problem. Let's go to Andrew. Would you say catabolic or anabolic tryptophan operon?
Anabolic.
It is anabolic. It makes stuff. The environmental tryptophan, signal tryptophan acts as a what? Erica Marston. Are you there, Erica? Are you able to unmute?
Oh, sorry, my space bar was not working for
Yeah.
unmuting myself. I'm thinking When I'm hearing tryptophan, I'm thinking of like a co-receptor.
Yeah, you're close. It's a co-repressor. That's right.
Oh, okay.
Co-repressor. Yep. Good job. By default, the repressor is active or inactive, Alina.
Are you asking me?
Yes. Is the repressor for the tryptophan operon by default, is it active or inactive?
I will pass.
That is not a problem. John, what would you say? John says active. That is not correct. But again, I think it is just this double negative thing. This is the most difficult question for everybody to answer. So now Mariceli can feel a little bit better. So thank you for saying that, John, because I bet she's like, oh, thank goodness it wasn't just me. By default, the repressor is inactive. Remember, because what we want by default is for transcription to be, let's have everybody weigh in here. Transcription for the tryptophan operon is by default on. That's right. Okay, so this is all filled in for you. Okay, good job, everyone. The last thing we need to talk about real quick is change. Okay, and this is very relevant to this week's lab. Now, when we think about genetic change, which is what drives evolution in living cells and in viruses too, genetic change can happen by mutation in all forms of life. That is when a cell goes through cell division or viruses go through replication and the genetic material is copied. Mistakes can be introduced that are inherited by the offspring. That's what happens in humans and that's what happens in bacteria. But there are additional mechanisms of genetic change that are much more rapid and dramatic in prokaryotes that don't really happen in eukaryotes for the most part, and that involves transduction, conjugation, transformation. And so these are considered to be horizontal forms of gene transfer. Vertical transfer is when you inherit DNA from your parents. Horizontal is when you receive DNA from a peer. So that would be like if I went and sat down at Starbucks and I was happy to be sitting next to some rando over here who was having a strawberry frappuccino and I accidentally got some of his DNA. So what are the names of those three horizontal gene transfer mechanisms? Again, I just mentioned them. Let's see if you remember them. Throw them into the chat real quick. So remember that mutation is the vertical one. Mutation is the one where we make small mistakes in the course of copying the genetic material prior to cell division. Transformation, conjugation, and transduction are the horizontal methods. We get them from non-relatives, basically. So for each one of them, I'm just going to ask if someone can raise your hand and define it. So for transformation, how would you define it? Raise your hand if you have a good definition of transformation. Feel free to use the words that I've used in the prep. Mariceli, go ahead.
Is that the naked DNA uptake by bacteria?
Yes, that is uptake of naked DNA from the environment. Good. What about conjugation? Raise your hand if you can define that. Aminata, go ahead.
It is a DNA transfer between bacterial cells.
Yes, and it's specifically between living cells, and they're physically connected to each other. And last but not least, transduction. Raise your hand if you have a good definition for that. Mariceli, go ahead.
Is that the bacterial DNA transfer between viruses?
Yes, viruses are the intermediate in that case. So the transformation we have, basically the bacteria finds some DNA lying around. Conjugation is when two living bacterial cells physically connect, and transduction is when they transfer DNA using a viral intermediate. So we're going to briefly talk about this experiment. Okay. This is one of those three mechanisms, and this is one of the first experiments in the early 1900s that demonstrated this was going on, and people were very shocked by it. So let me walk you through what's happening here. First of all, this is the pathogen Streptococcus pneumoniae that causes one form of bacterial pneumonia. You can see that it's encapsulated. That's one of its virulence factors, making it invisible to the mammalian immune system. If you inject it into a mouse, the mouse will die, and you can recover live cells in the blood that, when you look at them under the microscope, are basically this cell. There is a relative of this strain, so a mutant strain. It's got a mutation in the ability to make capsules, so that gene doesn't work. You inject those cells into a mouse and they can replicate. You can recover them and look at them and see they're the same, but the mouse does not get sick. You can also take these dangerous bacteria and heat kill them. If you inject that into the mouse, they don't replicate. You can't recover anything. The mouse stays healthy. But the crazy thing is that when you mix these two harmless reagents together, then what happens is these living cells seem to somehow turn into these heat-killed ones. Because these are like dead as dead can be. Either that or they are somehow reanimated. Like, what's going on? Because when you do this, you inject the mouse, it dies. And what you recover is a living form of the encapsulated bacteria. So what mechanism is demonstrated here? That's right. That's correct. This is transformation. And if you can explain in a little more detail how this worked, like how come this happened, raise your hand. Go ahead, Mariceli.
Did the cells just mutated?
So it is that these living cells did go through change. That is part of it. Nicole, what would you like to add?
Okay, so the encapsulated cells were killed, so they're dead. So the living non-encapsulated cells take the DNA from the dead ones by transformation, which allows them to make capsules and turn them into the dead ones in a way.
Yeah, that's exactly right. So great answers, both of you. Thank you. Exactly what happens when these heat-killed cells, or when these cells are heat-killed, they start to fall apart. And one of the things that's released is their DNA. These live cells come upon that DNA, and a small, tiny fraction of them are going to accidentally or just environmentally take up that DNA. That's going to give them the gene that they're missing to make capsules, and they basically turn into these bad ones. Okay, now bacterial conjugation, I told you, was living cells. So this is not a dead and alive cell situation. Both cells are alive. Conjugation is the closest equivalent to bacterial sex. They basically exchange DNA while they're still alive through some kind of physical connection. It is important to know that gram-positives and gram-negatives do this differently. Gram-negatives use a fertility pilus. So one cell will actually have the genes to make this long, hollow pilus, and it will poke another cell and inject its DNA into that cell. Gram-positives form a mating bridge between themselves, and that allows the DNA transfer to occur. So again, which cells use the pilus and which use the mating bridge? For the pilus, who's using that? Gram-positives or gram-negatives? Yep. And then that means, of course, the gram-positives are using a mating bridge. So one of the things that this should tell you is that conjugation cannot happen between cells that are different gram statuses. It's either gram-negative, or it's gram-positive, but you're not going to do conjugation between a gram-negative and a gram-positive. And then this mechanism we'll talk more about next week. This is when a virus infects a cell, and in the course of making more viruses, some of the bacterial DNA gets inside the capsid. And when it goes to target its next host, bacterial DNA from the previous host ends up in the new host. This is transduction. So we're gonna talk more about that next week and do a Kahoot. I do want to just remind you that you are doing the unknown ID project right now in step 3. So make sure to read the instructions, watch the video, and come to lab if you still have lab this week, or when you come next week, ready to ask any questions if you get stuck or you're feeling confused about this process. Please do not sit around procrastinating or feeling stressed or anxious because you don't know what to do. And don't do this project blindly, because that will cost you points at the end. I want to remind you what's due this week: genetics review quiz. And even though the official deadline for part 2 has passed, if you're one of the few people who hasn't completed it, please get that done. A reminder that lab 6, which is this week, is not yet due. We will not have data until next week, but there is an online component that you would do well to begin now, so you're not trying to do everything at the last minute. And this is super important, guys. So next week is the last subject in the Unit 2 material. And it is viruses, and it is fascinating, but there's more genetics, and it is really important that you do your prep, like super important. Watch those videos, read those slides, come to class with some background so that you can make the most out of these Zoom meetings that we have together. And don't forget to do pre-lab 7. Okay, so with that, I'm going to stop the recording and take any questions that you have.